periodically, and I'll do the best I can. If you have questions that I'll need to ask, but this is where we're at. So I'm gonna zoom in, and if I zoom in too much, we'll start to get problems. And if I zoom in not enough, then you can't see it. So if you need to move, feel free. I got seats up here. You can sit down here, and if I go anymore, we're gonna start doing the Skittles color stuff. So you don't just get glycolysis, you get to like view the rainbow. And that will start really soon. So again, less than ideal, we gotta make do, right? Nothing if not flexible. So here we go. Let's talk about glycolysis and where I had left off 5 minutes ago, which is we need to get this glucose into the cell. So I had mentioned before it froze on you, not on me, it kept going on me. But I'm gonna talk about getting this insulin to our insulin receptor and what happens next. So insulin, this is a hormone, and its job is to come out of the blood, diffuse through the ECM, and bind to this insulin receptor. Misconception number one, people think that insulin enters the cell. It does not. It has no business being in the cell, and the cell wouldn't know what to do with it anyway. Its job is to hit that receptor and activate that receptor, and that's it. Insulin's gonna come back off of that receptor and could easily go back into the blood. It does not enter the cell. Misconception number one. Does insulin enter the cell? The answer is clearly no. The cell would not know what to do with it. What insulin does instead is it activates this receptor, and through what's called a second messenger system, which I'm going to sidestep, it causes our glucose transporter, or GLUT, to be inserted in this membrane. GLUTs are not always present. They need to be told, now is the time to be present, and insulin does that. So the presence of insulin causes glucose transporters to be inserted. This is how we prevent cells from taking up too much glucose. Otherwise, they'd kind of be greedy. So if that glucose transporter was always there, that cell could always get glucose in from the blood, assuming the concentration gradient favored that. And we don't need that. Cells don't need that much glucose. It's wasteful. So the insulin tells the cell, now is the time. We have a lot of blood glucose. This person just ate. Blood glucose is high. Please take some of this. So this causes our glucose transporter to be inserted. So our glucose transporter, commonly known as the GLUT, is inserted into the cell membrane. So that means it can also be taken back down. Everything in the cell is very dynamic and responds to other needs in the body. So far, so good. So step one is glucose enters the cell via facilitated diffusion. So this is a reminder, facilitated diffusion means needs help getting into the cell, needs to be facilitated, and this GLUT is the protein that does that. So thing one, we get glucose into the cell. Now we're ready to start glycolysis. So we're going to assume everything went well. We have no endocrine issues, we had no diffusion issues, we had no facilitated diffusion issues. So we're going to start the process of glycolysis. And I'm going to move this over a little bit because when I walk, it'll pick up my shoes and that throws off the focus. We are nothing if not high tech here. So glycolysis. And when I talk about glycolysis, I'd like to zoom in a little more so you can see it. Or why not be brave? There we go. We're going to do something called follow the carbons. So if you had glycolysis in another course, which I'm sure many of you have, you were presented with a lot of different enzymes. And those truly are important, but I'm picking out the things that have a broader physiological context, as in they are helpful somewhere else in this class that we're gonna look at. Or they have a direct implication to a disease. So I'm not gonna hit all like 12 steps of glycolysis. I'm hitting the ones that are most important for us. So just that this looks different than what you've seen in the past, I understand that. My tip for you is to follow the carbons when it comes to figuring out what is glycolysis, what is its purpose, what does it look like, where or where not are we going to see carbon dioxide produced. follow the carbons. So can you remind me how many carbons are in a molecule of glucose? 6. So when we do glycolysis, I'm not worried about hydrogen or the oxygen in glucose. I'm just looking at the carbons because it's these carbon bonds that contain the electrons. And it's the electrons that actually fuel the electron transport chain. So we're following the carbons all the way through from the start of glycolysis to the end of the electron transport chain. It's all about the carbons. So we're going to start by just looking at like a simple carbon chain. This is what glucose looks like if we just looked at the carbons. So 6 carbons there. I would recommend you draw 6 dots because we're going to start to cleave these. We're going to start to add some phosphates to them. And I think if you sort of follow this linear path, it gets a little bit more interpretable and you don't feel quite so lost when you're studying glycolysis. So I've got 6 carbons here. So glucose in my parentheses there says 6 C's, as in 6 carbons. There's a lot of energy stored in between these bonds. These bonds here that hold the carbons together, these have a lot of electrons in them. And that's our goal, is to get those out, but get them out in a way that's methodical so we don't waste them. We also don't want to create too much heat. Enzymatic reactions always create heat, and cells are kind of picky about how hot they're going to get. So we have an engineering challenge. Acquire the energy here, but in a way that's not wasteful, and in a way that doesn't create too much heat. So that's a lot to manage, and cells do it quite well. So first of all, I'm going to introduce to you this really important enzyme called hexokinase. Hexokinase. I have underlined the root word here, which is kinase. And you will see this as a common theme kind of throughout lectures from this point forward, which is why I spend time talking about this root word called kinase. Kinases move phosphates. That is their job. If you know nothing else about the prefix, because there's different types of kinases, if you know nothing else about this enzyme, and we know it's an enzyme because it ends in ASE, enzymes end in ASE, so you already know a few things about this word without having to memorize, and that's helpful. If it ends in ASC, it's an enzyme. If it is kinase, we know it's an enzyme and it does something with a phosphate. It moves this phosphate around. So that's a lot of things you could use going forward. It helps you eliminate things that it doesn't do. So kinases move phosphates, enzymes that move phosphates. When we think about what kinase does for this glucose molecule, before I go any farther, if I don't alter this glucose molecule, there is nothing preventing it from leaving this cell if this glute is still available. Facilitated diffusion does not care which way things go. Doesn't care at all. So if we had glucose enter the cell and suddenly blood glucose got too low, if we didn't do something with our glucose molecule, it could diffuse right back out. And then this cell would be left with nothing. So luckily, cells are really smart and they have a plan. They have a plan. So this hexokinase is the first thing that's going to interact with glucose, and it's going to prevent this glucose from ever being able to leave via the glute that may still be expressed. So let's talk about what it does. Hexokinase, remember kinases add phosphates to something, or remove phosphates. So we still have, as a result of this hexokinase, we still have our 6 carbons here. So our 6 carbons here. But look what happened. We added a phosphate. Now we have glucose 6-phosphate. And now that seems kind of like a random detail, like why does that matter? But it matters a whole lot to the cell. Oh, sorry. This thing's super touchy. I'm trying to zoom in so you can see it, but we got like this weird wheel thing. I don't know. Let's just see. Let's just test it to failure. Will it fail? Yes, it will. Let's keep going. Okay. Okay, enough is here. Glucose 6-phosphate, that's what we get from hexokinase. This is a very different molecule according to the cell. Glucose 6-phosphate cannot go backwards. It cannot use a glute. So once we put this phosphate on our glucose, it's kind of locked into the cell. And that's really helpful because it prevents loss of glucose to the outside world. So hexokinase, hugely important for glycolysis. Some cells will just stop here. They'll add that phosphate and they'll store their glucose as glucose 6-phosphate. The liver is like that. And then when blood glucose does fall, when you're sleeping, for example, because you're not eating, the liver cells can cleave that phosphate, make glucose, and it can leave. So there's benefits of knowing sort of both ways of this. Let's see what happens next. So after hexokinase adds a phosphate to this, we're going to see that our glucose 6-phosphate is exposed to another enzyme, phosphoglucose isomerase. Phosphoglucose isomerase. Is it an enzyme? Yes, because it ends in ASE. What does an isomerase do? Think about the word isomer. Isomer. This is an enzyme that creates an isomer. What's an isomer? Bingo. Great job, Reese. Same molecular formula, sort of different orientation. And that has a really important thing that has to do with glycolysis. So with our phosphoglucose isomerase, just look at the word isomerase. It is an enzyme and it does something with an isomer. It makes an isomer. What we're going to do is we just rearrange some of these carbons. Now, our eyes, they look the same. We still have a line of 6 carbons. But if we could zoom in, we could see that the carbons switch places in some spots. And that makes this more likely or more sort of a propensity to split. Because we need this thing to split. We're sort of preparing this carbon chain to split in half. And this isomerase is our first sort of chance to get this to split in half. So phosphoglucose comes from here, isomerase, we rearrange these carbons. And when we do, we get a new compound, fructose 6-phosphate. Still 6 carbons, still the same phosphate added to carbon number 6. But the other carbons in this area have been rearranged and this thing is about to get unstable. We're going to see one more enzyme that's important because we can practice kinases. Phosphofructose, fructose phosphate, you can see how they get their name, kinase. Now, as I've mentioned before, kinases do something somewhat predictable. And it should be pretty obvious what that is if you're drawing this along. What did this kinase do? It added a phosphate because that's what kinases do. They move phosphates. Sometimes they stick a phosphate onto something, sometimes they remove a phosphate, but at the end of the day, what do kinases do? They move phosphates. And so we went from a 6-carbon beast that had 1 phosphate. Through the action of this kinase, we added another phosphate. So now we have 2 phosphates on either end. We've rearranged the carbons in the middle to make them unstable. What do you think's about to happen? I mean, look at this thing. Yeah, it's about to break apart. It has no other option. We've added some high energy molecules to each end. That makes it unstable. And as I mentioned before, we've rearranged the carbons here to make them unstable. Like, this thing really has no other option at this point but to split apart. And that's good. So we've been methodically sort of picking at it, provoking it, making it want to split apart. Before I continue, let me talk about its name, fructose 1,6-bisphosphate. These numbers, 1 and 6, tell you which carbons the phosphate is on. Carbon 1 has a phosphate. Carbon 6 has a phosphate. So these numbers don't have to be so mysterious. They're just being descriptive. If you don't really know how to interpret that, you're like, here's one more thing that I don't really know why we're calling it this. So fructose, carbon 1 has a phosphate, carbon 6 has a phosphate. So fructose 1,6-bisphosphate, that's its name. It's got a long name. So the next thing we're going to do is we're going to see an enzyme called aldolase. This is not a kinase, by the way, so we're not going to move any phosphates. Before I go any farther, how many ATP molecules does it take to make glycolysis happen? 2. Do you see how we've just done that? Now, some of you probably have it memorized. Okay, whatever. Or you could just look at this and go, well, we started with a chain of 6 carbons that had no phosphates. And now we have a chain of 6 carbons that has 2 phosphates. Where did that come from? ATP. So when we talk about it takes 2 ATP molecules to make glycolysis start, You can prove it to yourself. Don't take my word for it. Look at it. We just took 2 phosphates from ATP molecules. So we took 2 ATP molecules, and now we have 2 ADP molecules. At this point, if we stopped right here, this would be a terrible deal for the cell, because it would not have gained any energy. Instead, all we've done so far is use energy. But we're gonna start to get a payoff. So far, so good. Any questions? Quick questions that I can help you with? Clarifications, handwriting. So far, so good. Okay. I'm gonna keep going then, and we're gonna see that aldolase, you know it's an enzyme, ends in ASE. Aldolase is gonna split this thing apart. And here's where I start to get pretty abbreviated, because these steps are not as important to information in the future. I have 2 molecules. I've split them apart. They still have phosphates. So here's sort of the first one, and here's the second one. So I know it kind of looks crowded. Maybe you're like, I don't see 2 molecules, but there truly are 2 molecules, 3 carbons each, and they both have a phosphate on the 3rd carbon. I didn't really add anything to these. I just split them apart right down the middle to get 2 groups of 3 carbons, and the phosphates took some rearranging. These 2 molecules, 3 carbons each with a phosphate, and this looks like the same molecule. These are called G3P, glyceraldehyde 3-phosphate. The 3 tells you which number of carbon is it on the 3rd carbon. Glyceraldehyde 3-phosphate. Much easier to abbreviate as G3P. Glyceraldehyde 3-phosphate. Before I go any further, how many carbons are in each pyruvate molecule, which is the end of glycolysis? At the end of glycolysis, we get pyruvate. How many carbon molecules per pyruvate? 3. Do you see how we're about to get that? We're about to get some really great stuff. We've set ourselves up for success. We already have 2 molecules, 3 carbons each, just like pyruvate. And we're going to start to see what happens to these phosphates, but not yet. So we're just methodically getting ready for the end goal, which is making pyruvate and creating some ATP. Let's take a look at what happens next. We're going to actually add a couple more phosphates. A phosphate to this first carbon and a phosphate to this first carbon. So to our G3P molecules, one here, we add another phosphate. So now we have 3 carbons, phosphate on each end, and this molecule is called 1,3-bisphosphoglycerate. Again, the 1 and the 3 tell you where are the phosphates. They're on the first carbon and they're on the 3rd carbon. 1,3-bisphosphoglycerate. A really great molecule. This has a lot of benefit for the cell. Let's talk about how many ATP molecules does glycolysis net? 2. We're about to see how that happens. Right now, I have 4 phosphates. And you can probably predict from these 4 phosphates, I'm going to get 4 ATP molecules. We're going to take these phosphates off. We're going to add them to an ADP molecule. We're going to get ATP. So right now, we're going to make 4 ATP molecules. And so I could say, well, how many ATP are created during glycolysis? The answer is 4. But how much did it take to get it going? 2. So be careful with the wording here. Net means what is your sort of at the end of the day when you do your balance sheet, what did you gain? Well, we gained 4, but we put 2 in, so we only gained 2. So net is 2. But we're going to get these phosphates off. We're going to make ATP, and that process has a really interesting name, which I'm going to give you now. It's called substrate level phosphorylation. And this happens throughout glycolysis. We see it happen when we do the Krebs cycle. Substrate level phosphorylation. really fancy term for meaning, we're gonna take ADP that is in the cytoplasm, and with that ADP in the cytoplasm, we're gonna add a phosphate. So we take a phosphate off. I'm not good with this projector, this camera here. And as a result of adding phosphate, we get ATP. And we get ATP in situ, in place. So it's called substrate level phosphorylation. And I'm about to get 4 of them. But it took 2 to get it done. So far so good? Substrate level phosphorylation. How we can make ATP with glycolysis or Krebs cycle. Moving down, we have 2 pyruvates. So as I mentioned, I don't talk about glycolysis in the detail you probably saw it in biochemistry. They do a great job of it. I'm just hitting the highlights that are necessary for us now and will be necessary for us when we talk about skeletal muscle physiology. Because skeletal muscle cells have a lot of ways of making this happen in ways that other cells don't. So in a way, we sort of start with skeletal muscle physiology. What happens when you train an athlete really well for an athletic event? You're doing a lot of work behind the scenes cellularly. So 2 pyruvate. Notice pyruvate has no phosphates because of this. We took them off and we made ATP. So this is glycolysis for our purposes. We started with glucose, ended with pyruvate. In the process, we created some ATP. We also created something called NADH, reduced coenzymes that carry electrons. Questions before we continue? Yeah.
So I see that for like what phosphates do. The first 2 I added to like ATP. How would the second?
Ooh, great question. Where do those second set of phosphates come from? Those are free phosphates that are in the cytosol. So ATP is not the only thing that can donate phosphates to molecules. Creatine phosphate is another molecule that can donate. And we also just have free phosphates. I say that so carefully because I get tongue-tied. Free phosphates in the cytosol. So you're right, most phosphates do come from ATP, but not all. Not all. Yeah.
Is it possible that the first 2 phosphates added were the free, like from the cytosol?
The first 2 do come from ATP. Yeah, it's the enzyme reactions that are coming from G3P to 1,3-bis-phosphoglycerate. We're going to see an enzyme that takes a free phosphate that's in the cytosol and add that to it. Different reaction. Great question. Any other questions before I continue? Okay, I have some questions for you. 3 questions. Just looking at glycolysis. Looking at glycolysis. Okay. I don't want to go any smaller because then you won't be able to see the questions. In reference to your paper so far, have we produced any carbon dioxide? Why or why not? So the first part's easy. Did we make carbon dioxide? No. Why not? Second question. The conversion of G3P to 1,3-bis-phosphoglycerate produces 2 NADH. Checking to see if you were listening previously. What is the significance of NADH? And question 3. Pretty simple, but I just want to make sure we're all on the same page. What is the net gain of ATP from glycolysis? Net. Not gross. How do we produce overall? What do we end with at the end of the day if we did our balance sheet? I'm going to give you a couple minutes to work on this, and then I'm going to continue. Feel free to chat it up with a neighbor. These things will help you understand the next part of my lecture. So that's where I like to stop and say, how are we doing? Any questions on this? So work through this, and then I'll continue. realize I'm tied to the Bob Barker microphone. I hate that because I can't move. I don't like that. If you can hear me, that's good. If you can't hear me in the back, let me know. So let's talk about question 1. Has any carbon dioxide been produced yet? In glycolysis. Did you see any? No. And the answer is no. No, we haven't produced any carbon dioxide. Why not?
We still have all 6 carbons.
Bingo. Very good. We still have all 6 carbons. Carbon dioxide has a carbon. If you produce carbon dioxide, you must have removed a carbon from somewhere. We don't. We still have 6 carbons. We rearranged them and we split them in 2, but we didn't lose any. So no carbons were lost, no carbon dioxide produced, no oxygen needed. So when we talk about does glycolysis require oxygen, the answer is no, but a declarative statement is not very helpful. What is helpful is saying why. And it's because you don't have to remove any carbons. Therefore, you don't need oxygen, which does the dirty work of removing the carbons. So, why or why not? No carbons removed. No carbons were harmed in the making of this video. Question 2. Conversion of G3P, G-bisphosphoglycerate, produces 2 NADH. What does NADH do? What's the significance? What does it carry? Electrons. So let me talk about this for a moment in case it's been a while since you thought about NADH, which is sad because I bet NADH thinks about you all the time. And would ever such a case. So if I was to come over here and talk about NADH, I'm going to back up one step. NAD plus, no electrons, which I'm going to abbreviate E negative. That's a little abbreviation for electrons. No electrons, and it's probably really sad. Much like the fact that you don't think about it. It thinks about you all the time. You never give it the time of day. If we add electrons to this, I'm going to add 2 electrons. This thing is electron hungry. If I add 2 electrons to this, which we get from some of these reactions, we get a new compound called NADH. Sometimes people get fancy and say NADH plus H. I feel like that's extravagant. I'm just going to keep it simple, NADH. This has electrons. It accepted 2 electrons and it's pretty happy. It's done its job. These electrons are, think of them as little bits of energy that are liberated every time we break carbons apart, and their goal is to get to the electron transport chain, but they need help. Because right now, some of them were liberated in the cytoplasm. They have no way of getting into the mitochondria. There's dog hair in my mouth. It's an inappropriate time for that, but I don't get to choose. You dog people know, right? You don't get a choice. There's a dog hair. Anyway, we get NADH, 2 electrons on board. This is going to go to the inner mitochondrial membrane and help fuel ATP production. Like big time. So we want NADH to be produced. It is super important. When you talk about glycolysis, people really focus on substrate level phosphorylation, super important, but in my opinion, not nearly as important as this. Because that ATP we just created here, it has a lifespan of 2 or 3 seconds. ATP is a really short-term storage molecule. So that's not a big deal. But the electrons, they have a further job. Okay. So, next question, question 3. Net gain of ATP from glycolysis, what's the number? 2? 2, good job, 2. Because we got 4, but we took 2 to get it going. We are leaving glycolysis, and we're gonna move into the next part. This thing ate my markers. It was a good day earlier, you know, and then it just wasn't. So glycolysis, when you're studying this, break it down. If this is overwhelming for you, it should be. There's a lot of stuff. You're not alone. Break it down. Stop when you're like, I can't do this anymore. Fine, take a break. So we're gonna move into thing 3. And remember, this is a fluid thing. We just made some pyruvate, and now we're gonna move on and see why did we make that pyruvate? Was it just for the hell of it? No, we're gonna do something with it. So let's take a look at the next step. I'm gonna bring it up here. Pyruvate enters the mitochondria. So I've just brought the pyruvate to sort of the top of the page, that's all. I haven't changed it, I didn't do anything with it. I just brought our 2 molecules of pyruvate from down here to up here. And we have the next challenge, which is we have to get these molecules through a phospholipid bilayer. So this is a good time to practice diffusion. Do you think we can get these into or past the phospholipid bilayer just on their own? No, these require some help. Believe it or not, nobody really knows how this is done, which I think is kind of cool, right? Like you look at something like this and you think we have it all laid out. We don't. So I'm just going to give you what we know. This could change. But we have two different channels that are required to get this pyruvate just into the mitochondria. I'm going to give you a moment to write this out. I have two channels, different colors, different channels. These are going to be ways we get pyruvate into that mitochondria. Two different channels required. I'm also going to remind you of some mitochondrial anatomy. Mitochondria, interesting little organelle for sure. It has two different membranes. It has an outer and an inner membrane. And these matter a lot, not just in composition, but in what happens at these membranes and sometimes in between these membranes. So just take a moment. This is a fairly straightforward drawing here. We're going to be doing something with it in a moment, but right now we're trying to figure out how we're going to get that pyruvate into that mitochondrial matrix. It's in the cytoplasm. We need it inside this mitochondria. But as we decided, it can't enter with simple diffusion. It needs help. It's got 3 carbons. It's too big. So I'm going to talk about the top membrane, the outer mitochondrial membrane. It's going to have a channel. We're going to talk about the inner mitochondrial membrane that has another channel, and both of these are required to get pyruvate in. There are genetic diseases where these aren't produced correctly and a person cannot produce enough ATP simply because they can't get pyruvate into the mitochondria. It's a rare genetic disease, but they do exist, which actually drove the understanding of the mechanism. These people didn't have any relief because they didn't know what was causing it. Good scientists, good research, good biochemistry answered that for us. So we're going to see that this pyruvate is going to come through. So both of these do the same route. They're going to come through this outer mitochondrial membrane, so this inner mitochondrial membrane, and they're going to arrive at our inner mitochondrial matrix. So 2 different proteins. I'm not giving them names. I'm purposefully sidestepping that because in literature it's not well known. So all I need you to know is it takes 2 different proteins for the facilitated diffusion of pyruvate into the mitochondrial matrix. Now, let's talk about what happens next. So, I got my pyruvate, 2 of them, in the mitochondrial matrix. I'm just reminding you the inside part of this mitochondria is called the matrix. I haven't changed this pyruvate at all. All we've done is hopefully get it into the mitochondria. So far, same molecule. But we gotta do something with it. And that something is make some acetyl-CoA out of it. So we're going to see that pyruvate is about to undergo a change. I'm going to be going through just one of these. We realize I have 2 of these, so the reaction I'm about to show you would happen twice, but that's kind of redundant. I don't need to show it to you twice. So 2 molecules of pyruvate, and we'll get 2 molecules of acetyl-CoA at the end. Here's our reaction. We're going to start with pyruvate. We're going to take off a carbon. We're going to add a CoA, coenzyme A, and we'll get acetyl-CoA. So each pyruvate, 2 pyruvates, go through the same reaction. We get 2 molecules of acetyl-CoA. I'll let you write that up. And then I have some questions for you. Be careful with your carbons. About to have a change here. Alright, got this down? I'm seeing a lot of eyeballs. I have it written down on our page. Okay. What's the biggest difference between a pyruvate and an acetyl-CoA molecule? What's the biggest difference? Just look at the structure. We lost a carbon. We went from 3 carbons to 2 carbons. Everybody see that? We kicked off a carbon. And that's a big change. It looks small, but some physiological stuff is going to definitely happen as a result. So we take this carbon, and carbon cannot get out of the mitochondria. It couldn't even get out of the cell membrane on its own. So what do you think we're going to add to this carbon so that we can get it out? Oxygen. This is really the first time we see oxygen needed in any of these reactions. So I'm going to add some oxygen to this carbon, and now I get CO2. And it turns out CO2 can diffuse out through these phospholipid bilayers unaided. Simple diffusion, because we added some oxygen. When this carbon dioxide leaves the mitochondrial membranes, goes into the cytoplasm, it should leave the cell through the phospholipid bilayer, enter the ECF, and then the blood. What happens to the pH of the ECF or blood if we have too much of this accumulate? It goes down, becomes acidic. So here's sort of our first, hey, remember, this has big time sort of implications to the overall homeostasis of the person. Hopefully we're making ATP in such a controlled way that the CO2 we're producing is very easy to handle by the blood. The blood can take it, whisk it to the lungs, and you exhale it. Or the kidneys help get rid of it. But if you keep producing this faster than you can get rid of it in the blood, then we're going to have something called respiratory acidosis, which I mentioned on Tuesday. So here's our first sort of, hey, remember, this has broader implications. So other things that we can produce here and that I want to talk about include the production of NADH. So NADH here, we went from NAD plus to NADH. What did we pick up when we created this molecule? Electrons. We picked up electrons, and that is going to be huge. So, first question due to or related to acetyl-CoA production. When a carbon was removed from pyruvate, what did that carbon form? That should be fairly straightforward to answer. What was released from the carbon cleavage? Meaning when I took that carbon off of this 3-carbon molecule, what did I also liberate? First part of this question, fairly straightforward. When carbon is removed from pyruvate, what did we form with it? CO2. So the first part of this question is CO2, we hope, otherwise we can't get that carbon out. What also was released when we took this carbon off? So we went from 3 carbons, took one off. What was released or liberated? And the answer was, Benedicta has already volunteered this, electrons. Electrons, or E with a little negative sign, electrons. So not only have we created the substrate for the next reaction, which is Krebs cycle, we produced some NADH. We've captured some electrons. And remember, those are kind of the unsung heroes to this whole process, is the creation of electrons. So we're methodically breaking down this carbon chain, and every time we release a carbon, yes, we get carbon dioxide produced, but this is really what we are after. So far, so good. Any questions? Quick clarification types. Yes?
The heat and ATP also do release since you're breaking the bond.
So we're not making ATP here, no substrate level phosphorylation, but yes to heat. Yeah, we did not remove any phosphates. We didn't have any phosphates to remove. The carbons were sort of bare, I guess.
Okay.
Yeah. Any other questions for you continue? All right. Sort of your halfway point, your study break when you're studying this later. NADH is produced during glycolysis and during production of acetyl-CoA. What is its purpose? How many times have I asked this question already? And how many times will you see it on the exam? Wink, wink, right? Wink, wink. What are we creating? What's the purpose of NADH? Carry electrons. Every time you see this word, NADH, think electron taxicab. That's its purpose. And it could not have a more important purpose. The creation of ATP along the way, sort of a neat, gee whiz, bang factor, but not really that important later. It's like a sparkly, glittery thing. Ooh, neat, but who cares? So it's really the electrons that we're after. So I'm asking this question many different ways because it's just that important. So what is its purpose? We're trying to get electrons or carry electrons from sort of the site or source of liberation. And I realize that does sound awfully dramatic, but that's what they call it. So anyway, site of liberation to the site of need. Maybe this makes it sound more important if I'm over dramatic with it. Otherwise those electrons will be lost and we can't really do much with them. And we would make a lot of ATP subsequently. So I'm going to go from this, we're going to see why do we need to make acetyl-CoA. We produce 2 acetyl-CoA per molecule of glucose, and that's going to fuel the Krebs cycle. And the Krebs cycle has different names, citric acid cycle, for example. There's a third name for it, which we're using for now. If you've studied the Krebs cycle in the past, you know it is a big, hairy, long, awful thing. In this class, it's not that bad. It really isn't. So you're like, yeah, great, but truly, it's really not that bad. In fact, it looks like this. This is the Krebs cycle for our purpose. We're going to go from acetyl-CoA. We're going to combine that with oxaloacetate. We're going to get citrate. And for our purposes, this is all we need to know. There's some sort of important things, some details that I will add. Not enzymes, not who does what where. You really need to know these 3 things. The acetyl-CoA that we just produced over here are 2 carbon molecule acetyl-CoA. First thing it does is it combines with a molecule of oxaloacetate. So thing 1, oxaloacetate needs to be present. It's a cycle. We always have to regenerate oxaloacetate. 4 carbons. Acetyl-CoA had 2 carbons. So when I combine oxaloacetate and acetyl-CoA, I get 6 carbons. All I'm doing is just adding some carbons. 4 carbons plus 2 carbons, 6 carbons. And I form citrate, which is why sometimes this is called the citric acid cycle. Citrate is the first thing that's produced. It is unstable. So do we have this much on our paper? I don't want to continue if you don't, because there's no recording, it's hard to go back. We good with this? Yes. Notice citrate has 6 carbons. And the first thing we need to be aware of is it's called the Krebs cycle because we have to regenerate the starting point. The starting point is oxaloacetate, 4 carbons. Citrate, 6 carbons. Oxaloacetate, 4 carbons. What we're going to do in the real sort of meat of the Krebs cycle is we take the 6 carbons here, we take 2 of them off. We're going to talk about what we get when we remove them, but first realize we have to regenerate oxaloacetate. Our original glucose molecule has now been fully oxidized. There are no carbons left to work with. We've either taken them off and made carbon dioxide, got some electrons out of them, or we used them to regenerate oxaloacetate. But we didn't leave anything on the table. There's no waste. Everybody okay with that? Where is glucose fully oxidized? The end point, Krebs cycle. That's where we see the last carbons either removed from our original glucose molecule or incorporated back into our next oxaloacetate molecule. But there's nothing left. We used all the original carbons. When we take these carbons from citrate, what are we going to produce? We get 2 carbons, they can't get out. What must we add? Oxygen. So this carbon plus oxygen and this carbon plus oxygen, obviously we get CO2. So now we can get that carbon dioxide out, so we've dealt with that problem, which is, what do we do with these carbons that we just took off? So, got that down. What did we liberate when we cleaved those carbons? Electrons. What do you think we created with those electrons? NADH. And FADH2, which is the same purpose, slightly different molecule. So we got NADH, which we've been talking about the whole time, and also we're gonna get one molecule of FADH2, which also carries electrons. It's just like a taxi cab of a different color. Maybe one's a Chevy and one's a Toyota. They do the same thing, just slightly different. So when we look at what is the purpose of Krebs, per turn of Krebs, this is what we get, which means per acetyl-CoA molecule that enters, we get all this stuff. Krebs is a big deal. We get 3 NADH. There's 6 electrons there. We get 1 FADH2, 2 electrons there. A molecule of GTP, which is a high energy compound, very similar to ATP, and we got 2 carbon dioxide molecules. Remember, this is per turn, which means per acetyl-CoA molecule that starts. Per turn of Krebs. That's a lot of stuff. Krebs cycle is hugely, hugely important for fueling our last reaction of aerobic respiration. If it wasn't for Krebs, we'd have a really hard time making the next part work. Before I continue, question 6. Checking to see, did we retain this information? After Krebs, is glucose fully oxidized? What is the answer? Yes. All the 6 carbons that came from glucose have either been taken apart and released as carbon dioxide, or were used to regenerate oxaloacetate, but there is nothing left of our original 6 carbon molecule. So it's not until Krebs cycle that we can say glucose is fully oxidized. Oxidized just means we blew it apart. It done blew up. But like carefully, without too much heat or any waste. Alright, let's see what was the big deal with all of our reduced coenzymes. I'm going to introduce to you the last part of our electron transport chain. Our last part, which is the electron transport chain. Step 5, electron transport chain or oxidative phosphorylation. Electron transport chain and the production of many, about 32 molecules of ATP. About. Will I ask you numbers like that in the exam? No, because they change. Per person, per cell, per minute. Numbers are kind of irrelevant. But good for trends, I guess. Step 5. Electron transport and production of many ATP molecules. What you're looking at here is one set of complexes of the electron transport chain. And we're looking at the inner mitochondrial membrane. We have lots of these sets. I'm going to say they come in a set. We have complex 1, complex 2, complex 3, complex 4, and then the big deal at the end, ATP synthase molecule. So here is a set of complexes. And the inner mitochondrial membrane is full of these sets. Let's talk about what these complexes do. So complex 1, Roman numerals here. Here we see NADH converted or oxidized to NAD+. What do you think we gave up when we did that? Look at the difference between these two. What did we put in to make this? Electrons. So notice we don't have hydrogen over here. It turns out when we oxidize or sort of break this apart, we get 2 things. Electrons and hydrogen, because that's what went in to make it. So when we break it apart, we just get those parts back out. Electrons are going to stay in our respiratory chain, the electron transport chain. See if I can zoom in. Wrong button. There we go. So we're going to see little e, negative, electron. This is going to bounce around through the chain. And it provides energy, which takes the hydrogen from here, and it pushes it, it pumps it into this inner mitochondrial membrane space. So we've got outer mitochondrial membrane, inner mitochondrial membrane. We've got the space in between. The space is really important because this is where we collect hydrogen. So complex 1 strips electrons from this and hydrogen from this. The electrons bounce through this chain and they fuel the pumping of hydrogen into this space. Complex 2, we do the same thing, but with a different molecule. FADH2, also our electron taxichem. We're gonna take hydrogen from it, we're gonna take electrons from it. We oxidize it back to its original state. And we're gonna see here, we also pump hydrogen. Complex 3 is kind of a pass-through for these electrons. Complex 4 takes these spent electrons that have been bouncing through this chain. Complex 4 helps provide just enough energy to provide even more hydrogen in our inner mitochondrial membrane space. At this point, our electrons are pretty low energy and we have to pull them out of the chain, otherwise they'll back up this entire system. When we pull electrons out of this chain, we actually combine them, use them to combine oxygen and hydrogen, and we get water. Metabolic body water is produced by complex 4. By pulling spent electrons out of this chain, the spent electrons are used to join hydrogen and oxygen together, and we get water. Metabolic body water, complex number 4. Over time, as these chains work together, my last point before I let you go, we will have collected a ton of hydrogen, like just an absolute ton of hydrogen in this space. That's a form of energy, a source of energy. Hydrogen desperately wants out. It cannot get out though, because our outer mitochondrial membrane, our inner mitochondrial membrane, they're phospholipid bilayers and they're not permeable to things with a charge. Their only way out is through our ATP synthase molecule. It's like a huge flow of hydrogen through this molecule. And as our hydrogens flow through this molecule, that's what creates ATP. ATP synthase is really a little rotor stator. As this hydrogen flows through here, it turns a little molecular motor. And that creates enough energy to phosphorylate or stick a phosphate on, going from ADP to ATP. And that's where I will end, 'cause I'm out of time.